Okay, let's unpack this. Imagine a hidden language, written not with letters but with sugars, silently shaping everything from how a virus infects you to how your body fights disease and even how a single sperm finds and fertilizes an egg. It's a world we rarely think about, a molecular symphony bustling with activity right inside our cells, orchestrating functions we often take for granted. Today, we're embarking on a truly fascinating deep dive into glycoproteins, these, well, unsung heroes of biochemistry. You sent us some incredibly detailed source material, and our mission today is to break down every concept, every intricate mechanism, and every surprising implication of these complex molecules. We're going to go deep, exploring the nuance and the interconnectedness so you can truly master this information like the elite professionals in this field. Get ready for some serious aha moments because what these molecules do is truly astonishing.
Indeed. What's fascinating here is that we'll be exploring how these molecules, which are essentially proteins elaborately decorated with unique sugar chains, are involved in a vast and fundamental array of biological processes. We'll look at their fundamental structure, dissect how they're meticulously built and precisely broken down, and critically uncover their profound importance in both maintaining health and driving disease. From the smallest viral attachment to the complex regulation of our cellular identity, glycoproteins are absolutely key players.
We'll cover everything from the basic what are they to the intricate details of their synthesis pathways, how they act as crucial biological information carriers, their surprising roles in conditions like, say, diabetes and rheumatoid arthritis, and even how they're exploited by insidious pathogens like the influenza virus and HIV. It's a truly foundational topic with far-reaching consequences that touch almost every corner of human biology. All right, let's peel back the layers then. When I first heard glycoprotein, I immediately pictured something like, a protein with a sugary coating, pretty straightforward. But the source material hints that the definition is much more nuanced and precise. What's the fundamental truth about what these molecules really are, beyond just the simple name?
You've hit on a crucial point there. If we connect this to the bigger picture, glycoproteins aren't just any protein with a sugar attached. They are proteins that specifically contain oligosaccharide chains, which we call glycans, and these glycans are covalently bound to specific amino acids within the protein. This attachment of sugars, known as glycosylation, isn't some minor afterthought. In fact, it's the most frequent and functionally diverse post-translational modification of proteins in our bodies. This means the protein is first fully synthesized based on the genetic code, and then after its initial creation, these complex sugar decorations are precisely added. This entire process is highly enzymatic, meaning it's carefully controlled by a multitude of specialized cellular machinery. It's a deliberate and essential modification that dictates much of a protein's final form and function.
And you highlighted a critical distinction earlier in the material, glycosylation versus something called glycation. For someone just dipping their toes into this, what's the key distinction that separates these two processes and why does it matter so much? They sound so similar, it could easily be confusing.
Absolutely. This is a critical distinction that often causes confusion, yet understanding it is fundamental to grasping their biological significance. Glycosylation, as we just established, is a sophisticated enzymatic process. Think of it as a highly controlled, purpose-driven cellular construction project, meticulously managed by a vast array of enzymes called glycosyltransferases. It's precise, deliberate, and vital for normal cellular function. Glycation, on the other hand, is a completely non-enzymatic process. It's a spontaneous chemical reaction where sugars, primarily glucose, randomly attach to amino groups on proteins, and even to DNA or lipids, without any enzymatic control or cellular regulation. Imagine it as an accidental sticky mess rather than a deliberate decoration. While glycosylation is essential for life, allowing proteins to function correctly, glycation can have severe pathological consequences. This is particularly evident in conditions like poorly controlled diabetes mellitus, where consistently high blood glucose levels accelerate these non-enzymatic reactions, leading to widespread cellular and tissue damage throughout the body. It's the difference between a finely tuned biological symphony and, well, random chemical noise.
So it sounds like the difference between a cell's deliberate, intricate construction of a sugar signal versus an accidental sticky mess from too much sugar. That's a powerful core insight. Given this, it seems these molecules are absolutely everywhere. The sources they occur from bacteria to humans and even in viruses. What are some of their broad functions? They truly sound like the silent workhorses underpinning so many processes.
Precisely. This subtle distinction between controlled enzymatic precision and uncontrolled chemical chaos is a fundamental battleground in our biology. As for their ubiquity, you're absolutely right. They are incredibly versatile, performing numerous functions across all forms of life. Their carbohydrate content can vary wildly, from a mere 1% to over 85% by weight, which immediately tells you about their incredible structural and functional diversity. They act as fundamental structural molecules like the various collagens that give our tissues strength and integrity. They also serve as powerful lubricants and protective agents, like mucins, which form robust physical barriers on delicate epithelial surfaces, from our airways to our digestive tract. Many crucial transport molecules in our blood, such as transferrin, which carries iron, and ceruloplasmin, which carries copper, are glycoproteins. Immunological molecules, including antibodies, immunoglobulins,
Yeah.
and histocompatibility antigens, which are essential for our immune system to recognize self versus non-self, are also glycoproteins. They can even function as potent hormones, like chorionic gonadotropin and thyroid-stimulating hormone, or TSH, or as enzymes, such as alkaline phosphatase, which plays a role in bone metabolism. Furthermore, they are absolutely critical for cell attachment and recognition, influencing how cells interact in tissues, how viruses and bacteria bind to host cells, and even how hormones interact with their specific receptors. And here's a surprising fact from the source. They can even act as anti-freeze proteins in cold water fish plasma, preventing ice crystal formation. So think of it this way. If proteins are the workhorses and DNA is the blueprint, glycans are the secret sauce that dictates how, when, and where those horses perform, and how that blueprint is truly expressed in real time. It's the dynamic, intricate layer of control we're only just beginning to fully decode.
Wow, that's an incredible range of functions, from antifreeze to immune defense. And what's particularly intriguing is that they're not static, are they? The source hints at their dynamic nature, changing in response to cellular signals, which is quite astounding for molecules that also provide structural integrity.
Correct. The glycan structures of glycoproteins are not fixed. They are remarkably dynamic, constantly adapting. They change in response to signals involved in critical biological processes, like cell differentiation, where a cell commits to a specific fate. Normal physiological adjustments, like wound healing or immune activation, and even pathological conditions such as neoplastic transformation, cancer development. This dynamic alteration in their sugar structures is a direct result of different expression patterns of glycosyltransferases, those specific enzymes responsible for adding sugars to proteins. These enzymes dictate which sugars are added, in what sequence and at which locations, thereby shaping the final glycan structure. This constant adaptation and rewriting of the sugar code isn't just theoretical. It's a fundamental mechanism by which cells respond to stress, grow, and unfortunately even turn cancerous. This raises an important question. If the structure can change so dramatically and in response to cellular cues, what kind of information are these intricate sugar chains actually encoding? How does this sugar code actually work?
This is where it gets really interesting. The source states that these oligosaccharide chains encode biological information. How does this sugar code differ from the genetic code we usually hear about, the one in DNA and RNA, which seems much more direct and, well, immutable?
This is a critical distinction and a core concept for mastering this topic. The information in glycans is considered secondary information, unlike the primary information encoded in DNA, RNA, and proteins, which is directly template-driven. The genetic code is precise and follows a strict linear template, ensuring exact replication and protein synthesis. It's like a pre-written book. The sugar code, however, is far more nuanced, more like a dynamic interactive symphony where the outcome depends on the entire cellular environment. The specific pattern of glycosylation on a given protein depends on several highly variable factors within the cell. Firstly, it hinges on the expression pattern of the various glycosyltransferases present in a specific cell type or even a specific cellular compartment at a given moment. Different cells or cells in different states will express different sets of these enzymes, creating unique glycosylation fingerprints. Secondly, it relies on the affinity of these different glycosyltransferases for their particular carbohydrate substrates, how strongly they bind and react with specific sugars, which can influence which pathways are favored. And thirdly, it's influenced by the relative availability of the different carbohydrate substrates, the sugar building blocks themselves, within the cell. Because of these multiple dynamic variables, most glycoproteins display what we call microheterogeneity. This means that not all glycan chains on a given protein are identical. There can be variations. Some chains might even be truncated or incomplete compared to others on the same protein, or even on the same type of protein in different cells or at different times. This inherent variability isn't a flaw, but rather part of its sophisticated nature, allowing for incredible fine-tuning and responsiveness.
So how is this secondary information actually read or expressed by the cell if it's not a direct template? It sounds like a more subtle, perhaps more adaptable form of communication.
You're right, it's a subtle but profoundly powerful form of communication. based on molecular recognition. The information for these sugars is expressed through highly specific interactions between the glycans themselves and other proteins or molecules. The most prominent example, which we'll delve into shortly, are lectins. These are specialized carbohydrate-binding proteins, essentially nature's sugar detectives. When a glycan binds to a specific lectin, or another complementary molecule, this molecular recognition event doesn't just sit there. It triggers a cascade of changes in cellular activity. These changes can range widely, from modulating a protein's solubility, viscosity, and overall three-dimensional conformation, to providing crucial binding sites for everything from other cells to invading pathogens. Glycans also provide vital cell surface recognition signals, allowing cells to identify each other and interact in structured ways, protect underlying proteins against premature degradation by proteases, and ensure the correct folding and precise targeting of proteins to their proper destinations within the cell. They can even protect circulating hormones and plasma proteins from premature clearance by the liver, acting as a vital quality control mechanism. So it's a dynamic, highly responsive signaling system that relies on precise molecular recognition, influencing a vast array of cellular processes, constantly adapting and relaying information.
If glycans are chains of sugars, what are the common sugar building blocks we find in human glycoproteins? Are there hundreds of them, or a select few that the cell cleverly rearranges to create this diversity?
That's a great question, and it's another aha moment. Despite the astonishing complexity of glycan structures, only 8 monosaccharides predominantly appear in human glycoproteins. This is a remarkably small set of building blocks, yet the combinatorial possibilities of how these 8 can be linked, branched, and arranged allow for an astonishing diversity of structures, far greater than that achieved by even a large number of amino acids and proteins. These 8 are galactose, galac, glucose, GLC, mannose, man, and acetylneurominic acid, NUAC, fucose, FUC, N-acetylgalactosamine, GalNAc, N-acetylglucosamine, GLC-NAC, and xylose, Dixyl. To give you a sense of their typical arrangement and function, N-acetylneurominic acid, which is a type of sialic acid, is almost always found at the very end or termini of oligosaccharide chains. It acts like a cap or a crucial recognition point, typically attached to subterminal galactose or N-acetylgalactosamine residues. The other 7 sugars are more commonly found in internal positions within the chain, forming the backbone and crucial branching points that define the glycan's unique shape. Additionally, sulfate is often found attached to these sugars, usually to galactose, N-acetylglucosamine, or N-acetylgalactosamine, adding further complexity, negative charge, and modulating their biological activity, such as in immune recognition or cell adhesion.
And are these sugars added directly to the growing chain, straight out of the cellular soup, so to speak, or do they need some kind of special preparation or activation first?
No, they don't just get picked up directly. It's not the free sugar or even just a phosphorylated sugar that's the direct substrate for glycoprotein synthesis.
Yeah.
Instead, it's the corresponding sugar nucleotide forms. Think of these as activated energy-carrying versions of the monosaccharides, like specific delivery trucks for each sugar, such as UDP galactose, GDP mannose, or CMPN acetylneurominic acid. These nucleotide-linked sugars provide both the necessary energy required for the formation of the glycosidic bond, the chemical linkage that joins sugars together, and act as the precise delivery system for the sugars to be incorporated into the growing glycan chain. This activation process ensures that sugar addition is an energetically favorable and highly regulated reaction, crucial for building these incredibly complex structures accurately and efficiently. Without these activated forms, the intricate assembly line of glycosylation simply wouldn't work.
That's a great overview of the fundamental pieces, the building blocks, and their activated forms. Now let's move on to some of the molecular players that interact with these fascinating sugar structures, specifically those sugar detectives you mentioned earlier, the lectins.
Indeed. What's fascinating here is the crucial role of lectins as primary readers of the sugar code. Lectins are a diverse group of carbohydrate-binding proteins that possess a remarkable ability to agglutinate cells, essentially clump them together, or to precipitate glycoconjugates, which are molecules like glycoproteins that have carbohydrates covalently attached. It's important to note a specific distinction here. Immunoglobulins, or antibodies that react with sugars, are generally not considered true lectins, as their primary function is immune recognition, not direct carbohydrate binding in the same way. A key characteristic that defines a lectin's function in agglutination or precipitation is that it must contain at least 2 sugar binding sites. A protein with only a single sugar-binding site simply can't cross-link and clump cells or precipitate glycoconjugates, highlighting the importance of multivalency for their specific biological actions. It's like having two hands to grab and pull things together.
Where do we find these lectins, and can you give us an example of their function beyond just clumping cells? What's their practical significance in our bodies?
Lectins were first discovered in plants and microorganisms, which is why plant lectins were formerly called phytohemagglutinin due to their prominent ability to clump red blood cells. But we now know many animal lectins are vital to our own biology, playing roles in immunity, cell adhesion, and protein turnover. A prominent example that demonstrates their sophisticated function is the mammalian Escherichia glycoprotein receptor, found predominantly in the liver. What's fascinating here is how the liver uses this receptor for a crucial aspect of protein turnover and quality control in our bloodstream. If a plasma protein loses its terminal N-acetylneuramic acid, a process called desilylation, which often happens as proteins age or get damaged, the underlying galactose residue is exposed. The Escherichia glycoprotein receptor on liver cells specifically recognizes this newly exposed galactose moiety. This recognition isn't just passive, it triggers the rapid clearance of that desilylate protein from circulation, usually through endocytosis, where the liver cell engulfs the protein, and subsequent catabolism, breaking it down. This elegant quality control mechanism ensures that old or damaged plasma proteins are efficiently removed, preventing their accumulation and maintaining physiological balance in the blood.
So on a more practical everyday note, undercooked beans could be an issue then if they contain these potent plant lectins. It sounds like they're not just benign plant compounds.
Indeed, it's a powerful reminder that even seemingly innocuous molecules can have profound biological effects when they're not handled correctly, whether in the lab or the kitchen. Undercooked legumes containing undenatured lectins, such as phytohemagglutinin in kidney beans, can be problematic for human consumption. These potent plant lectins can actually agglutinate the mucosal cells lining our intestines, leading to stripping of the intestinal lining, which can cause significant gastrointestinal distress, including nausea, vomiting, and diarrhea. This highlights their potent biological activity and why proper cooking, which denatures these proteins, is so important before consumption. Beyond their physiological and pathological roles, lectins are also incredibly valuable research tools in biochemistry and cell biology. They are used to purify specific glycoproteins from complex mixtures, to probe the glycoprotein profiles present on cell surfaces, providing insights into cell identity and state, and even as reagents for generating mutant cells that are deficient in specific enzymes involved in the biosynthesis of oligosaccharide chains, which helps us study sugar metabolism and its pathways. They allow scientists to literally read the sugar code on cell surfaces.
The source material now breaks glycoproteins down into three main groups based on how the sugar chain is linked to the protein. Let's unpack these fundamental structures and what makes each class distinct, because this is where the diversity really begins to shine.
This raises an important question. What defines these major classes and allows for such incredible functional diversity? It's a precise nature of the covalent linkage, the specific chemical bond between the polypeptide chain and the oligosaccharide. The source illustrates these clearly in its diagrams. There are O-glycosidic linkages, where the sugar is attached to a hydroxyl group, typically on serine or threone. N-glycosidic linkages, where it's attached to an amide of nitrogen, specifically on asparagine. And glycosylphosphatidylinositol, or GPI linkages, which anchor proteins to membranes via a lipid tail. While these are the three major classes, accounting for the vast majority of human glycoproteins, there are also other minor types. It's also crucial to remember that a single protein can even contain more than one type of sugar chain. For example, glycophorin, an important red cell membrane glycoprotein, contains both O and N-linked oligosaccharides, showcasing the complexity. The number of oligosaccharide chains attached to one protein can vary widely, from just 1 to 30 or more. And the chains themselves can range from a couple of sugar residues to much larger, incredibly complex branch structures, creating an almost infinite array of possibilities.
Let's start with O-linked glycoproteins. Where do we find these, and what's unique about their structure and the specific types of O-glycosidic linkages that define them?
O-linked glycoproteins are characterized by a direct covalent linkage between the hydroxyl side chain of either serine or threonine amino acids, and sometimes, though less commonly, tyrosine and a sugar, most commonly N-acetylgalactosamine, forming what's known as a Galnac-Surther linkage. This is the predominant O-glycosidic linkage found in human glycoproteins, although the source outlined at least 4 distinct subclasses that serve different purposes. Beyond this predominant linkage, for instance, in proteoglycans, which are giant, highly glycosylated molecules, we find a galactose galactosylose trisaccharide, often called the linked trisaccharide, specifically attached to serine residues. In collagens, which are crucial structural proteins, there's a unique galactose hydroxylysine linkage. And interestingly, many nuclear and cytosolic proteins have a single N-acetylglucosamine sugar attached to a serine or 309 residue. This last type of O-glycosylation with just a Secoul sugar is particularly dynamic and involved in rapidly reversible metabolic regulation, almost like a molecular switch, which we'll definitely discuss further.
And a prime example of O-linked glycoproteins is mucins, right? They sound pretty important for protection and lubrication, but how do their O-linked structures give them these properties?
Mucins are a truly fascinating and vital class of O-linked glycoproteins that perfectly exemplify how structure dictates function. They are highly resistant to proteolysis, meaning they are incredibly difficult for enzymes to break down because the dense arrangement of their oligosaccharide chains creates a formidable physical shield. This makes it challenging for proteases to access and cleave the underlying polypeptide chain. This remarkable resistance is crucial for their protective roles. They serve vital functions in lubrication and in forming a robust physical barrier on delicate epithelial surfaces, such as those lining our respiratory, gastrointestinal, and reproductive tracts. Imagine them as a slippery protective coat that keeps our internal surfaces hydrated and safe.
What makes mucins so special structurally that gives them these properties, particularly their high viscosity and gel-forming abilities?
Mucins possess two distinctive structural characteristics that underpin their unique properties. First, they have an incredibly high content of O-linked oligosaccharides, often comprising more than 50% carbohydrate by weight. This massive sugar decoration isn't just for show, it's key to their function. Second, their polypeptide chains contain variable numbers of tandem repeats, or VNTRs, of specific peptide sequences. These repeats are typically rich in serine, threonine, and proline, the very amino acids to which the O-glycan chains attach in dense clusters. The source even notes that up to 60% of our dietary threonine requirement can be accounted for by the synthesis of mucins, highlighting their sheer abundance in the body. This high density of O-glycans confers an extended, rigid, rod-like structure to mucins, almost like a stiff brush. This rigidity is partly explained by steric interactions between the N-acetylgalactosamine moieties and adjacent amino acids, which essentially stiffens the chain. Furthermore, the high content of negatively charged N-acetylnuraminic acid and sulfate residues, commonly found in many mucins, contributes significantly to their high viscosity and gel-forming properties, enabling them to form the thick, protective mucus we're familiar with. Mucins can be secretory, forming the soluble mucus, or membrane-bound. What's fascinating here is that membrane-bound mucins can also mask cell surface antigens, effectively hiding a cell from immune recognition. Cancer cells often exploit this mechanism, forming large amounts of mucins on their surface to evade immune surveillance. Conversely, some mucins also carry cancer-specific peptide and carbohydrate epitopes, which researchers are actively trying to exploit to stimulate an immune response against cancer cells, effectively turning a shield into a potential therapeutic target.
That's a clever evasion tactic by cancer cells, turning their own protective coating into a disguise. How are these O-linked chains actually built within the cell? It sounds like a highly organized and complex assembly line, especially given the specificity required.
Their synthesis is indeed a precise, stepwise process that highlights the cell's remarkable organizational capabilities. Since most glycoproteins are destined to be membrane-bound or secreted, their mRNA is typically translated on membrane-bound polyribosomes, meaning protein synthesis occurs at the endoplasmic reticulum, or ER. The glycan chains are then meticulously built up by the sequential addition of sugars. Each sugar unit is donated by its corresponding sugar nucleotide, those activated energy-carrying forms we discussed earlier. This entire process is catalyzed by a highly specific group of enzymes called glycoprotein glycosyltransferases. Many of these glycosylation reactions primarily occur within the lumen of the Golgi apparatus, a distinct organelle in the cell. This means there must be specialized carrier systems known as permeases and transporters to transport the sugar nucleotides, like UDP galactose, GDP mannose, and CMPN acetylnuraminic acid, from the cytosol, where they are synthesized, across the Golgi membrane into its lumen where they are needed. These transporters often function as antiporter systems, where the influx of one sugar nucleotide molecule is balanced by the efflux of one molecule of the corresponding nucleotide, like UMP, GMP, or CMP, ensuring efficient movement and maintaining balance. Humans possess remarkable 41 different types of these glycoprotein glycosyltransferases, organized into families and subfamilies based on the specific sugar nucleotide donor they use and the particular linkage they form. These enzymes show an incredibly high degree of specificity for their acceptor substrate, typically acting only on the product of the preceding reaction, creating a very ordered assembly. The different stages of glycan formation, and thus the different glycosyl transferases involved, are spatially separated within different regions of the sus-Golgi, medial Golgi, and trans-Golgi networks, providing a highly organized assembly line, ensuring the correct sequence of additions. Despite this precision, microheterogeneity can still occur, meaning some chains might be truncated or slightly varied due to enzyme availability or substrate concentration. Unlike N-linked glycosylation, no clear consensus amino acid sequence is known to universally determine which specific serine and threonine residues are O-glycosylated. But the first sugar moiety incorporated is almost always N-acetylgalactosamine.
Next, N-linked glycoproteins. If O-linked glycoproteins are like the protective armor and lubrication for our cells, N-linked glycoproteins, which the source tells us are the predominant class, seem to be the primary communicators and networkers. What makes them so dominant, and what are their major types that set them apart structurally?
That's a great analogy. N-linked glycoproteins are indeed the predominant class, encompassing a vast array of both membrane-bound proteins and circulating glycoproteins that act as key communicators. What sets them apart is their specific covalent linkage. The amide nitrogen of an asparagine residue forms a bond to an N-acetylglucosamine sugar. While they all share a common core pentasaccharide, which is a consistent structure of 3 mannose residues and 2 N-acetylglucosamine residues, MAN3-GLC and AC2, bound to asparagine, they then branch out into 3 major classes, differing significantly in their outer branches, which we often refer to as antenna. These classes are complex, high mannose, and hybrid.
Can you elaborate on these different types of antenna and how they distinguish the classes? It sounds like a cellular identifier.
Certainly. These antennas are indeed crucial for cellular identity and function. Complex oligosaccharides are incredibly branched and diverse, having anywhere from 2, 3, 4, or even 5 outer branches, or antenna. They generally feature terminal N-beta-cellular neuraminic acid residues, with underlying galactose and N-acetylglucosamine residues. These latter 2 sugars often form repeating N-acetylactosamine units, which can be quite extensive and are involved in many recognition events. Examples of these complex structures include the Thai blood group substances, which are found on red blood cells and define blood types. There's a bewildering number of complex type chains, with variations in branching in terminal sugars like galactose or fucose, making them highly specific. High-mannose oligosaccharides, as their name clearly suggests, primarily contain additional mannose residues linked to the core pentasaccharide, typically ranging from 2 to 6 extra mannose units beyond the core 3. They're simpler in their branching, but equally vital. Lastly, hybrid molecules combine features of both. They might have a complex type chain on one arm of the core and a high-mannose chain on another, representing a partial processing of the original structure. This diversity in branching and terminal sugars allows them to carry an enormous amount of biological information.
This sounds like an incredibly intricate assembly process, especially if it starts with such a specific core that then branches out. How does the cell manage to build these complex N-linked structures, particularly with that initial pre-assembly step?
The biosynthesis of N-linked glycoproteins is indeed quite involved and distinct from O-linked synthesis, highlighting a truly remarkable feat of cellular engineering. It doesn't begin with sequential sugar addition directly to the protein. Instead, the cell first synthesizes a large pre-assembled branched oligosaccharide structure. This sugar tree is initially attached to a unique lipid molecule called dolichol pyrophosphate, essentially fatty anchor. This initial assembly takes place on the cytosolic side of the endoplasmic reticulum, or ER membrane. Once formed like a complex modular unit being built off-site, this entire dolichol pyrophosphate oligosaccharide is then translocated, or flipped, across the membrane into the lumen of the ER. Here it undergoes further glycosylation, adding more sugar units, before the entire oligosaccharide chain is transferred in mass by a specialized enzyme called an oligosaccharyl transferase onto a specific asparagine residue of the nascent protein. Crucially, this transfer happens cotranslationally, meaning it occurs as the protein is entering the ER during its synthesis on membrane-bound polyribosomes. This makes it an efficient, all-at-once delivery system, crucial for creating these complex structures. In many N-linked glycoproteins, there's a consensus sequence, as L-next earther, where X can be any amino acid except proline, that helps guide this attachment, though not all sites strictly follow this exact sequence.
And the details of that initial dolichol-linked oligosaccharide synthesis are quite specific, aren't they? It's not just a random assembly. There's a defined pathway for building that initial sugar tree.
No, it's a very precise biochemical pathway, showcasing the cell's meticulous planning. The very first step involves UDP-N-acetylglucosamine reacting with dolichol phosphate, forming N-acetylglucosamine dolichopyrophosphate. Then, a second N-acetylglucosamine is added, again from UDP N-acetylglucosamine. Following this, 5 molecules of mannose are sequentially added, each donated from GDP mannose. At this point, the growing dolichopyrophosphate oligosaccharide have a core structure of 2 N-acetylglucosamine and 5 mannose units. This intermediate is then translocated or flipped from the cytosolic side to the luminal side of the ER membrane. Inside the ER lumen, the assembly continues. Additional mannose and glucose molecules are added to complete the pre-assembled oligosaccharide. What's distinct here is that these later mannose and aldoglucose residues are donated by dolichol P mannose and dolichol P glucose, which are also lipid-linked sugars, acting as internal carriers. This creates the final pre-assembled dolichol pyrophosphate oligosaccharide, a large structure with 2 N-acetylglucosamine, 9 mannose, and 3 glucose units. Once this entire block is formed, it is then transferred as a whole unit to the acceptor asparagine residue on the nascent protein chain. It is being synthesized and threaded into the ER. Following this bulk transfer, the chains undergo a series of trimming and further modification steps. For high mannose chains, specific glucose residues and some peripheral mannose residues are removed by glycosidases. For complex chains, all the glucose residues and 4 of the mannose residues are removed, both in the ER and Golgi, and then other sugars like N-acetylglucosamine, galactose, and N-acetylneurominic acid are added in the Golgi apparatus by specific glycosyltransferases, creating the branched antenna. Hybrid chains are formed by partial processing, resulting in a mix of complex and high mannose features on different branches of the same core. It's an intricate dance of addition and subtraction.
So it's not just about building them, but also about making sure they're built correctly and that the protein they're attached to folds properly. How does the cell ensure quality control for these intricate and linked glycoproteins, especially in the endoplasmic reticulum where so much protein folding happens? It almost sounds too complex to manage without constant errors.
That's an excellent point, and it raises an important question about protein folding and cellular integrity. It's true, it would be a chaotic mess without robust quality control. Glycoproteins undergo a rigorous quality control process within the ER, partly facilitated by specialized chaperone proteins like calmexin and its soluble counterpart, calreticulin. Calnexin, which is itself a lectin sugar-binding protein, plays a central role. It recognizes specific carbohydrate sequences within the glycan chain of the glycoprotein specifically, a monoglycosylated core where the terminal glucose residue has been removed, leaving only the innermost glucose attached. When a glycoprotein is in this specific state, calnexin binds to it, effectively preventing the glycoprotein from aggregating with other partially folded or misfolded proteins. Calnexin forms a crucial complex with ERP57, which is a key partner in this process, helping to ensure the correct folding by fine-tuning the disulfide bonds within the protein, which are vital for its three-dimensional structure. Once the glycoprotein is correctly folded, the sole remaining glucose residue is hydrolyzed by a glucosidase, causing the glycoprotein to be released from its complex with calnexin ERP57. At this point, if it's correctly folded, it's available for secretion or transport to its proper destination. However, if the glycoprotein is still misfolded, a specific glucosyltransferase enzyme acts as a folding sensor. It recognizes the misfolded state and reglucosylates it, adding back a glucose residue. This reglucosylation allows the misfolded protein to rebind to calnexin and the ERP57 complex, essentially giving it another chance to fold correctly. This cycle continues until correct folding is achieved. If a protein is persistently misfolded and incapable of achieving its native conformation, the system recognizes it. It is then translocated out of the ER back into the cytosol where it is tagged for degradation and catabolized. This entire elaborate system ensures that only correctly folded and functional glycoproteins proceed through the secretory pathway, preventing the accumulation of harmful misfolded proteins.
Wow, that's an incredible level of cellular intelligence we're talking about for a quality control system, constantly monitoring and correcting. And how is this whole complex process of N-linked glycosylation regulated within the cell? What dictates which sugar goes where and when?
The glycosylation of glycoproteins involves an astonishingly large number of enzymes. In fact, about 1% of the entire human genome codes for genes involved in protein glycosylation, which speaks to its immense biological importance and the sheer complexity of the machinery involved. This includes at least 10 distinct N-acetylglucosamine transferases and multiple isoenzymes for other glycosyltransferases, allowing for incredible specificity and fine-tuning. Key regulating factors in the first stage of N-linked glycoprotein biosynthesis. The assembly and transfer of the dolichopyrophosphate oligosaccharide include not only the availability of the specific sugar nucleotides, those delivery trucks for sugars, but also the presence of suitable acceptor sites on the proteins themselves, the precise tissue concentration of the oligophosphate, the lipid carrier, and the finely tuned activity of the relevant oligosaccharide protein, transferase enzyme. What's fascinating here is that even subtle changes in these regulatory factors can directly lead to pathologies. For instance, various cancer cells are often found to synthesize dramatically different oligosaccharide chains than normal cells, frequently exhibiting greater branching and altered terminal sugars. This alteration is due to cancer cells expressing different patterns of glycosyltransferases, often as a result of specific gene activation or repression within the cancerous state. These differences in oligosaccharide chains on the cell surface can profoundly affect adhesive interactions between cancer cells and the normal surrounding tissue cells, potentially contributing significantly to their aggressive behavior, including their ability to metastasize or spread to other parts of the body. It's like they're putting on a new, more invasive sugar disguise.
And then we come to the third major class, the GPI-anchored glycoproteins. These sound quite unique, especially with their membrane attachment. How do they compare to the other types, and what special tricks do they offer the cell?
These are indeed a unique and highly mobile class of membrane-bound proteins. They are uniquely anchored to the lipid bilayer not by spanning it, but by a glycosylphosphatidylinositol, or GPI, tail. This GPI linkage is actually the most common way by which a wide variety of proteins are anchored to cell membranes, highlighting its fundamental importance across diverse cell types. These proteins are specifically attached to the outer leaflet of the plasma membrane or the luminal leaflet of secretory vesicles via the fatty acids of phosphatidylacetol, which is a key component of cell membranes. Unlike transmembrane proteins that span the entire membrane and are relatively fixed, GPI-anchored proteins are essentially tethered to one side, giving them distinct advantages.
And their structure sounds quite elaborate, more than just a simple lipid anchor. There's a whole complex carbohydrate and phosphate structure involved.
It is indeed quite elaborate, far more than a simple fatty acid chain. The core structure involves the phosphatidylinositol itself, which is linked via N-acetylglucosamine to a complex glycan chain. This glycan chain typically contains various sugars, including mannose and glucosamine, forming a specific and often diverse carbohydrate sequence. In turn, this oligosaccharide chain is linked via a phosphorylethanol moiety, a small phosphate-containing group with an amino acid and an amide linkage to the carboxyl terminal amino acid of the protein. Furthermore, many GPI structures contain additional constituents that add to their diversity and tailor them for specific functions. For example, the GPI structure described in the source, which links acetylcholinesterase to the red blood cell membrane, contains an extra fatty acid attached to the inositol and an additional phosphorylethanol moiety attached to one of the mannose residues in the glycan. This allows for significant structural variations among different GPI anchors, creating a customized molecular tether for each protein and its specific function.
So what here? What unique functions do these GPI anchors provide that other membrane anchors or transmembrane domains don't? Why would a cell choose this complex anchoring mechanism?
They provide three key and distinct functions that are critical for cellular processes, which is why the cell invests in such a complex structure. First, and crucially, the GPI anchor allows for greatly enhanced mobility of a protein within the plasma membrane compared to proteins that employ transmembrane domains. Since the GPI anchor is attached only to the outer leaflet of the lipid bilayer, the protein is much freer to diffuse laterally within that fluid environment, unlike a protein that's embedded through both layers of the membrane. This enhanced mobility can be incredibly important in facilitating rapid responses to cellular stimuli, allowing receptors to quickly cluster or disperse as needed, for example, when responding to a growth factor signal. Second, some GPI anchors may directly connect with signal transduction pathways. This means that proteins that lack their own intrinsic transmembrane domain, meaning they don't directly span the membrane to signal inside, can, by virtue of their GPI anchor, nevertheless serve as functional receptors for hormones and other cell surface signals, effectively transmitting information into the cell. It's a clever way to relay messages. Third, GPI structures can specifically target proteins in either the apical or basolateral domains of the plasma membrane in polarized epithelial cells. This precise targeting is crucial for maintaining cellular organization, specialized functions like absorption or secretion, and overall tissue integrity, ensuring that specific proteins are located exactly where they're needed for directional processes. They are literally guiding proteins to their correct cellular addresses.
And how are these complex GPI anchors synthesized and then attached to their proteins? Is it another pre-assembly and transfer similar to N-linked glycans?
Exactly. Like the N-linked glycans, the GPI anchor is not assembled directly on the protein. Instead, it is preformed in the endoplasmic reticulum as a complete unit. The protein itself, which is destined to receive a GPI anchor, has specific targeting signals encoded within its amino acid sequence. Its primary translation product, meaning the protein fresh off the ribosome, has an amino-terminal signal sequence that directs it into the ER during its synthesis. Crucially, it also possesses a carboxy-terminal hydrophobic domain that acts as the specific signal for the attachment of the preformed GPI anchor. The synthesis of the GPI anchor begins with the insertion of the fatty acids of phosphatidylinositol into the luminal face of the ER membrane. This is followed by a series of glycosylation steps, starting with the esterification of N-acetylglucosamine to the phosphate group of PI. The glycan chain is then meticulously built up with other sugars like mannose and glucosamine, similar to the O and N-linked additions. A terminal phosphethanolamine moiety is then added to the completed glycan chain. In a crucial enzymatic reaction called a transamination, the hydrophobic carboxy terminal domain of the protein is precisely cleaved off. This is then immediately displaced by the amino group of the ethanolamine on the GPI anchor, forming a stable amide linkage between the GPI anchor and a specific aspartate residue in the protein. It's a very precise and elegant swapping mechanism that occurs after the ribosomal synthesis of the protein is complete, ensuring the protein gets its correct membrane attachment.
This is truly fascinating. The idea that glycosylation can be rapidly reversible, almost like a metabolic switch, especially with that single N-acetylglucosamine sugar. It implies a level of dynamic control we don't always associate with these complex structures.
What's fascinating here is precisely that rapid reversibility. Many diverse proteins, including critical components like nuclear pore proteins, proteins of the cytoskeleton, powerful transcription factors that regulate gene expression, proteins associated with chromatin, and even crucial nuclear oncogene proteins and tumor suppressor proteins undergo rapidly reversible O-glycosylation with a single N-acetylglucosamine sugar. This isn't a complex glycan chain, it's just one sugar acting as a regulatory tag. Crucially, the serine and threonine sites on these proteins where this OGL-CNAC is attached are often the same sites that are also subject to phosphorylation. This means that glycosylation and phosphorylation occur reciprocally in response to cellular signaling. It's a sort of molecular tug-of-war or a toggle switch, where one modification can displace or prevent the other, providing a finely tuned, fast-acting regulatory mechanism for protein function.
So 1 minute it's phosphorylated, the next it's O-glycosylated, depending on the cellular state and immediate needs. What enzyme manages this switch, and how does it achieve such a precise exchange, effectively replacing one chemical tag with another?
The key enzyme managing this switch is O-linked N-acetylglucosamine transferase, sometimes abbreviated as OGT. This remarkable enzyme uses UDP-N-acetylglucosamine as the sugar donor to have the single GLC-MSC. Intriguingly, it's been shown that OGT also possesses some phosphatase activity, meaning it has the capability to directly remove a serine or threonine phosphate group and immediately replace it with an N-acetylglucosamine. This dual reciprocal activity allows for a direct and rapid modification, ensuring a tight coupling between these two major regulatory pathways. While there's no single absolute consensus sequence that universally dictates where OGL-CNAC will attach, about half of these reciprocal glycosylation and phosphorylation sites occur within a specific amino acid sequence, often a pro-valser motif. The activity of this O-linked N-acetylglucosamine transferase is itself activated by phosphorylation in response to signals like insulin action, further integrating it into vast cellular metabolic networks. Conversely, the N-acetylglucosamine can be removed by another enzyme, N-acetylglucosaminidase, which then leaves the site available for phosphorylation again. This dynamic addition and removal of a single sugar provides a fast, highly responsive, and energetically efficient mechanism of metabolic control, essentially acting as a cellular rheostat, fine-tuning protein activity based on the cell's metabolic state.
And the source mentions this has significant implications for nutrient sensing and even various diseases, which makes sense if it's such a fundamental metabolic switch. How does it link to conditions like diabetes?
Indeed, the activity and the peptide specificity of O-linked N-acetylglucosamine transferase depend critically on the intracellular concentration of UDP-N-acetylglucosamine, its sugar donor. This is where the nutrient sensing comes in. A significant portion of glucose metabolism, specifically about 2% to 5%, flows through the hexosamine pathway, which directly leads to the formation of N-acetylglucosamine, the substrate for OGT. This metabolic link gives the O-linked N-acetylglucosamine transferase a direct and powerful role in nutrient sensing within the cell. The enzyme essentially reads the availability of glucose and other nutrients through the concentration of its sugar substrate. What's concerning is that excessive O-glycosylation with N-acetylglucosamine, which can lead to reduced phosphorylation of target proteins due to the reciprocal nature of these modifications, is directly implicated in key pathologies. It's thought to contribute significantly to insulin resistance and glucose toxicity in diabetes mellitus, where cells become less responsive to insulin signals in the presence of persistently high glucose. Furthermore, this dysregulation is also implicated in the progression of various neurodegenerative diseases, where disruptions in OGLC and ACT levels can affect neuronal function and survival. This truly highlights the profound importance of this delicate balance and dynamic regulation of single-sugar O-glycosylation.
You mentioned glycation earlier as a non-enzymic process with pathological consequences, quite distinct from glycosylation. Let's delve deeper into advanced glycation end products, or AGEs, and their role in the devastating complications of diabetes. It sounds like they're directly related to that accidental sticky mess analogy.
Glycation, as we distinguished earlier, is fundamentally distinct from glycosylation because it is a spontaneous non-enzymatic reaction that happens without the guiding hand of enzymes. It involves sugars, predominantly glucose, attaching to amino groups of proteins, but also to other crucial molecules including DNA and lipids. The process begins innocuously. Initially, glucose forms a reversible shift base with the free amino terminus or lysine residues of a protein. This shift base then undergoes a more stable, but still reversible, amidori rearrangement to form ketoamines. These ketoamines, over time and through further complex and largely irreversible reactions, accumulate and lead to the formation of what we call advanced glycation end products, or AGEs. The overall series of these reactions is widely known as the Maillard reaction, which might be familiar to you from the delicious browning and flavor development in certain foodstuffs during storage or heating, like the crust on bread or the caramelization of onions. However, in the human body, especially when blood glucose is consistently high, these same chemical reactions have much more severe implications, turning beneficial browning into destructive damage.
So how exactly do these AGEs cause damage, particularly in a condition like diabetes where blood sugar is consistently high? Is it just a physical alteration or is there more to it?
In poorly controlled diabetes mellitus, persistently elevated blood glucose levels lead to a significantly increased rate of glycation of various proteins throughout the body. When long-lived structural proteins like collagen, which is incredibly abundant in the extracellular matrix of almost all our tissues, or other proteins with slow turnover rates are glycated, their intrinsic properties are profoundly altered. For example, glycation can dramatically increase the cross-linking of collagen fibers, making tissues stiffer, less elastic, and impairing their normal function. Imagine trying to stretch a piece of stiff old leather instead of supple new material. This increased cross-linking and the accumulation of glycated proteins can lead to the retention and buildup of various plasma proteins, including low-density lipoprotein, LDL, often called bad cholesterol, in the walls of blood vessels. This accumulation in the arterial walls contributes significantly to atherogenesis, which is the process of hardening and narrowing of the arteries, a major and devastating complication in diabetes. Ultimately, AGs are strongly implicated in both microvascular damage, affecting small blood vessels and organs like the kidneys, leading to kidney disease, and eyes, leading to retinopathy and blindness, and macrovascular damage, affecting large blood vessels, contributing to heart attacks, strokes, and peripheral artery disease. These are all characteristic and devastating complications of diabetes mellitus.
And it's not just structural damage, is it? Cells seem to read or recognize these AGEs and respond in ways that contribute to inflammation, creating a feedback loop of damage.
They absolutely do. This is a critical aspect of age-mediated pathology, turning a simple chemical modification into a cellular danger signal. Certain cell types, such as endothelial cells that line our blood vessels, and macrophages, which are crucial immune cells involved in inflammation and tissue repair, possess specific AGE receptors on their surfaces, often called RAGE, receptor for advanced glycation end products. When glycated proteins, or AGEs, bind to and are taken up by these receptors, it doesn't just stop there. It triggers potent intracellular signaling pathways. Specifically, it can activate the transcription factor NF-κB, which is a master regulator of inflammatory and immune responses. Activation of NF-κB then triggers the generation and release of a wide variety of cytokines and other pro-inflammatory molecules, further exacerbating inflammation. This contributes to a state of systemic inflammation and further tissue damage, creating a vicious cycle in diabetic complications. The body, in trying to deal with these altered proteins, paradoxically ends up causing more damage through an overzealous inflammatory response. It's like an alarm bell that keeps ringing even after the fire has been put out, causing more destruction.
And the most well-known and clinically useful example of this Non-enzymic glycation in a clinical setting is the HbA1c test, isn't it? It's become a cornerstone for managing diabetes.
Precisely. The non-enzymic glycation of hemoglobin A, the main protein found within our red blood cells responsible for oxygen transport, leads to the formation of HbA1c. While this glycation occurs to a small extent normally in everyone, it is significantly increased in diabetic patients with poor glycemic control due to their consistently elevated blood glucose concentration. Since red blood cells have a relatively long lifespan of about 120 days, the amount of HbA1c in the blood reflects the average blood glucose level over the preceding 2 to 3 months, offering a valuable snapshot of long-term blood sugar control. As the source mentions, measuring HbA1c has become a very important and widely used part of the clinical management of patients with diabetes mellitus, providing a valuable long-term indicator of their blood sugar control and the effectiveness of their treatment, far more informative than a single fasting glucose reading.
Beyond structure and regulation, glycoproteins are involved in a huge array of biological processes that are fundamental to life itself. Let's look at some specific examples from the source, starting with something as foundational as human fertilization, which relies on incredibly precise molecular interactions.
Indeed. What's fascinating here is the incredibly precise and sequential role of glycoproteins in sperm-oocyte interaction, illustrating their vital function in reproduction. For a sperm to successfully fertilize an oocyte, it must first navigate and penetrate the zona pellucida, or ZP, which is a thick, transparent, and protective extracellular envelope that surrounds the oocyte. A key player in this initial recognition and binding is ZP3, an O-linked glycoprotein found on the zona pellucida. ZP3 acts as a specific, high-affinity sperm receptor. When a complementary protein on the surface of the sperm interacts with the oligosaccharide chains of ZP3, it triggers a crucial transmembrane signaling event within the sperm. This interaction induces the acrosomal reaction, a process where enzymes like proteases and hyaluronidase, along with other contents stored in the sperm's acrosome, a cap-like organelle, are rapidly released. The liberation of these enzymes allows the sperm to digest a path through the zona pellucida and reach the plasma membrane of the oocyte. Following this, another glycoprotein called pH30 becomes crucial. It's involved in the specific binding of the sperm plasma membrane to that of the oocyte and the subsequent fusion of the two membranes, which ultimately enables the sperm's genetic material to enter and fertilize the oocyte. This entire delicate sequence, from initial contact to membrane fusion, relies on precise glycoprotein recognition and interaction.
And what about their critical role in immunity and inflammation, specifically with selectins, which are involved in how our white blood cells get to where they need to be, almost like a cellular handshake for immunity?
Leukocytes, our white blood cells, are vital players in a multitude of inflammatory and immunological responses throughout the body. A critical first step in many of these responses involves transient interactions between circulating leukocytes and the endothelial cells that line our blood vessels. This interaction is absolutely necessary before the leukocytes can exit the bloodstream and migrate into the inflamed or infected tissues, a process called extravasation. Here, cell surface lectins called selectins play absolutely key roles in facilitating this intracellular adhesion. Selectins are single-chain, Ca2 plus binding transmembrane proteins, and their amino-terminal region contains a specific lectin domain that is expertly designed to bind to specific carbohydrate ligands on other cells.
How does this binding lead to an inflammatory response, from a seemingly gentle rolling cell to a full-blown immune attack? It sounds like a complex sequence of events.
It's a meticulously choreographed, multi-step process. In the initial phase, selectins on the surface of a leukocyte, for example, a neutrophil, bind with relatively low affinity to specific glycoproteins on the surface of activated endothelial cells lining the blood vessel. This transient binding causes the neutrophils to temporarily trap and roll along the endothelial surface, effectively slowing them down from the fast flow of blood. This rolling is essential for enabling the next steps. During this rolling, the neutrophils become activated by other signals, undergoing crucial changes in their shape and activating other stronger adhesion molecules on their surface, such as integrins. They then adhere firmly to the endothelium through stronger, more stable interactions between these activated integrins on the neutrophils and specific immunoglobulin-related proteins on the endothelial cells. After this firm adhesion, the neutrophils extend pseudopodia, which are temporary protrusions, into the junctions between endothelial cells. They then squeeze through these tight junctions, cross the underlying basement membrane, and are finally free to migrate into the extravascular space directly into the inflamed tissue where they are urgently needed. Selectins bind specifically to sialylated and fucosylated oligosaccharides, and even to some sulfated lipids, demonstrating the exquisite specificity of the sugar code. This precise binding specificity offers intriguing therapeutic possibilities. Administering compounds or monoclonal antibodies that block these selectin-ligand interactions could be therapeutically useful to inhibit excessive or harmful inflammatory responses, for instance in autoimmune conditions or reperfusion injury. This raises an important question. If these adhesion mechanisms are so important for immunity, what happens when they go wrong or when cancer cells hijack them for their own nefarious purposes?
And the source mentions cancer cells using these adhesion mechanisms, too, for their spread and invasiveness. It's unsettling how they can co-opt normal biological pathways.
Yes, unfortunately. Cancer cells often modify their cell surface glycoproteins and express specific selectin ligands on their surfaces. These altered surface molecules may play a direct and crucial role in their aggressive behavior, particularly in their ability to invade surrounding tissues and to metastasize, meaning to spread to other parts of the body, by interacting with selectins on endothelial cells in a manner analogous to how leukocytes attach. This allows them to adhere to blood vessel walls at distant sites, exit the bloodstream, and form new secondary tumors. It's a key mechanism in the spread of many cancers.
So when the synthesis or even the degradation of these incredibly precise glycoproteins goes awry, we see a whole host of severe diseases. Can you walk us through some of the most striking examples from the source material showing how critical these molecules are?
Absolutely. The intricate nature of glycoprotein synthesis and degradation means that even subtle defects can have profound and devastating consequences. One rare but severe condition is leukocyte adhesion deficiency type 2, or LAD2. This is caused by genetic mutations affecting the activity of a specific Golgi-located GDP fucose transporter. The critical result is a systemic absence of fucosylated ligands for selectins on the surface of white blood cells. This leads to a marked decrease in neutrophil rolling and adhesion, severely impairing their ability to reach sites of infection. Patients suffer from life-threatening recurrent bacterial infections due to their compromised immune response. They also typically exhibit psychomotor and mental retardation, highlighting the systemic role of fucose. Interestingly, some cases may respond to oral fucose supplementation, demonstrating a direct link between the metabolic defect and the symptoms. Then there's paroxysmal nocturnal hemoglobinuria, or PNH. This is an acquired anemia characterized by hemoglobin in the urine due to the hemolysis or breakdown of red blood cells. This hemolysis occurs particularly during sleep, reflecting a slight drop in plasma pH, which increases susceptibility to complement system lysis. The root cause is the acquisition of somatic mutations occurring after conception in hematopoietic stem cells. These mutations affect the enzyme responsible for linking glucosamine to phosphatidylinositol in the GPI structure. This leads to a profound deficiency of GPI-anchored proteins on the red blood cell membrane. Specifically, two crucial protectors, decay accelerating factor DAF or CD55 and CD59, are deficient. Without them, the complement system attacks the red cells unchecked, causing hemolysis and the symptoms of PNH. This understanding led to the development of life-changing complement inhibitors.
And some muscular dystrophies are directly linked to problems with glycans, which is surprising for a disease we typically associate with muscle structure. How do these sugar defects manifest in such a severe muscular disorder?
Yes, that's a very important connection, revealing the widespread impact of glycan defects.
Yeah.
Some congenital muscular dystrophies are, in fact, the result of defects in the synthesis of glycans on a crucial protein called isosteroid. This protein protrudes from the surface membrane of muscle cells and plays a vital role in connecting the muscle cell to its surrounding extracellular matrix by interacting with laminin 2 in the basal lamina. This interaction is absolutely essential for maintaining muscle integrity and stability. If the glycans of isosteroid are not correctly formed due to mutations in the genes encoding some of the specific glycosyltransferases responsible, this critical interaction becomes defective. This defective interaction then leads to the progressive muscle weakness, degeneration, and damage characteristic of muscular dystrophies. It's like having a crucial anchor point that's not properly secured. And in rheumatoid arthritis, we see another fascinating example of altered glycosylation contributing to disease, specifically involving circulating immunoglobulin G or IgG molecules. Specifically in rheumatoid arthritis, these IgG molecules often lack lactose in their SC regions and consequently terminate in N-acetylglucosamine. Mannose-binding protein, MBP, a lectin synthesized by liver cells, normally play the key role in innate immunity by binding to sugars on pathogens. In rheumatoid arthritis, MBP can bind to these agalactosyl IgG molecules, essentially flagging them as damaged because of their altered sugar tags. When MBP binds these altered IgG molecules, it subsequently activates the complement system. This activation then sets off a cascade of inflammation within the synovial membranes of the joints, leading to the chronic pain, swelling, and progressive joint damage characteristic of rheumatoid arthritis. It's like friendly fire by the immune system. What's interesting about MBP is its broader role. The deficiency in MBP due to mutation can make young infants highly susceptible to recurrent infections, further highlighting its importance in immune defense.
So many diverse diseases. What about issues with glycoprotein degradation itself, leading to harmful accumulation similar to some other well-known genetic disorders?
You've hit on a crucial point that brings us full circle. The proper turnover of glycoproteins is just as important as their synthesis. This involves the systematic catabolism or breakdown of their oligosaccharide chains by specific lysosomal hydrolases. Lysosomes are the cell's recycling centers. Genetic defects in the genes encoding these enzymes lead to abnormal degradation and the harmful accumulation of partially degraded glycoproteins within tissues. This causes a group of devastating disorders known as lysosomal storage diseases. The source lists several: monocytosis, fucosidosis, sinulodosis, aspartoglucosaminuria, and Schindler disease, each linked to a deficiency in a specific lysosomal hydrolase needed to break down specific glycan parts. This accumulation has severe consequences and exemplified by inclusion cell or eye cell disease.
Tell us about eye cell disease. That sounds particularly devastating, especially if it's about a faulty address label for these crucial enzymes.
Eye cell disease, also known as mucolipidosis 2, is a rare but extremely severe condition characterized by progressive psychomotor retardation and other physical signs, often leading to death in the first decade. The fundamental problem is truly fascinating. Cells from these patients lack almost all of their normal lysosomal enzymes within the lysosomes. As a result, lysosomes accumulate many undegraded molecules, forming characteristic inclusion bodies. What's crucial here is that the enzymes are synthesized normally. The problem isn't production, but their failure to reach the right destination, the lysosomes. Instead, they are incorrectly secreted into the plasma. The underlying issue lies in a faulty address label system. Lysosomal enzymes normally carry a specific mannose 6-phosphate recognition marker, like a postal code, guiding them to the lysosome. Patients with eye cell disease lack the Golgi-located N-acetylglucosamine phosphotransferase. This is the critical enzyme responsible for adding that mannose 6-phosphate tag. Without this essential tag, the enzymes can't be sorted correctly by the mannose 6-phosphate receptors in the Golgi.
Consequently, instead of being packaged and transported to the lysosome, they're simply secreted out of the cell. This defect in trafficking leads to the severe accumulation of undegraded material inside the lysosomes, causing devastating cellular and systemic pathology.
This is perhaps one of the most critical applications for many of us. How do glycans play a role in infections, particularly with pervasive viruses and bacteria? It seems like they're often used as invasion gates by pathogens.
You're absolutely right. Glycans are fundamentally involved in how many microorganisms bind to and infect human cells. Their ability to bind specifically to host proteins and other glycans is key to their pathogenesis, often the very first step. Take influenza virus A. It initiates infection by binding to specific cell surface glycoprotein receptors containing N-acetylneuromenic acid. This binding is mediated by its hemagglutinin protein like a grappling hook. But the story doesn't end there. Influenza also has a neuraminidase enzyme, which is crucial for allowing newly synthesized progeny viruses to elute or detach from infected cells after replication. If this elution is inhibited, the spread of the virus is significantly diminished. This understanding directly led to inhibitors like zanamivir and oseltamivir, which target this viral neuraminidase and are now used to treat influenza. This raises an important question. Given the constant mutation of viruses, how does our immune system keep up, and how do pathogens continue to evade our defenses?
And HIV-1, the cause of AIDS, also famously uses glycoproteins for its entry mechanism, right? Making them a key target for therapy and vaccine development.
Indeed. Human immunodeficiency virus type 1, HIV-1, relies heavily on its surface glycoproteins for cellular entry. It attaches via GP120, which binds to the CD4 receptor and co-receptors on target immune cells. Once attached, it uses another glycoprotein, GP41, to fuse the viral membrane with the host cell membrane, allowing viral entry. Antibodies against GP120 do develop, and there's been interest in using GP120 as a vaccine. However, a major challenge is that GP120's structure, particularly its glycan shield, changes rapidly due to mutations. This allows the virus to continuously evade neutralizing antibodies, posing a significant hurdle for effective vaccine development. It's a constant evolutionary battle, with glycans at the heart of the conflict.
And bacteria use them too, often for adhesion to host tissues, which is often the first critical step in establishing an infection.
Many bacteria absolutely do, exploiting glycans for essential adhesion. For instance, Helicobacter pylori, the major cause of peptic ulcers, meticulously binds to at least 2 different glycans on stomach epithelial cells. This allows it to establish a stable attachment, colonize persistently, and cause chronic inflammation. Similarly, many bacteria causing diarrhea attach to intestinal mucosal cells via glycans in glycoproteins or glycolipids. Even parasites, like the malarial parasite Plasmodium falciparum, use glycans, specifically a GPI present on their surface, to mediate attachment to human cells, a critical step in their life cycle. This truly highlights glycans as critical points of interaction for a wide range of pathogens, making them crucial targets for understanding and treating infectious diseases. So what does this all mean? We've taken a deep dive into the fascinating and incredibly complex world of glycoproteins, uncovering their precise definition as proteins with one or more covalently linked carbohydrate chains. We've explored their diverse structures, from single O-linked GLC and ACT to intricate branched oligosaccharides. We've detailed the 8 principal sugars and the indispensable role of sugar nucleotides. We thoroughly explored how these oligosaccharide chains encode biological information as secondary signals, modulating protein properties, and providing vital binding sites. We've delved into the 3 major classes O-linked, N-linked, and GPI-linked, understanding their unique linkages in complex biosynthesis, particularly the role of dolichol and the meticulous quality control by chaperones like calnexin in the ER.
And we haven't just scratched the surface. We've seen how remarkably dynamic these molecules are, from reversible glycosylation acting as a metabolic sensor to the dangerous non-enzymic glycation contributing to tissue damage in diabetes via AGEs. We also discovered their vital roles in fundamental processes like fertilization with ZP3 and in immunity, where selectins orchestrate leukocyte adhesion. Most crucially, we detailed how abnormalities lead to serious diseases like leukocyte adhesion deficiency, PNH, certain muscular dystrophies, and devastating lysosomal storage disorders like eye cell disease, where that faulty address label causes chaos. Perhaps most immediately relevant, we saw how viruses and bacteria like influenza, HIV, and H. pylori cleverly exploit glycoproteins to gain entry, making these intricate sugar structures critical therapeutic targets. It truly highlights how this sugar code impacts our health at every level.
This raises an important question. Given the astonishing complexity and absolutely critical roles of glycoproteins in every aspect of health and disease, and considering how their secondary sugar language is so finely tuned yet susceptible to subtle changes, what incredible breakthroughs in medicine and biotechnology might we unlock as we continue to decipher this intricate glycan code, moving beyond the genetic blueprint to fully understand and manipulate the nuanced, dynamic language of sugars? The potential is truly immense.
That's a powerful thought to leave you with, hinting at a future where we can perhaps rewrite or correct these sugar codes for profound health benefits. We hope this deep dive has not only informed you, but also ignited a deeper curiosity about the incredible molecular world within us. Thank you for joining us on this journey into the surprising and essential world of glycoproteins. Keep digging, keep learning, and we'll catch you on the next deep dive.